Hi all, welcome to Organic Chemistry Lab. This semester you'll be working with myself, Ray, and Tara to learn commonly used organic lab techniques this semester. Before I get started, I'm going to talk about general safety, and we're going to start off with lab attire. This is something you all have learned, but just a refresher, this is non-ideal lab wear. So Tara is coming in with her hair down and a baggy sweatshirt. Both of these are flame hazards and are very dangerous in the lab. However, she is wearing closed-toe shoes and pants, which is ideal. Tara has now changed her outfit. She has a t-shirt on and a few extra items. You can see she has a pair of goggles, which are always necessary, a lab coat, which is optional, and a hair tie to keep her hair out of the way. If you are wearing a scarf, that is acceptable, however it must be tucked in so it is not a flame hazard. Also, the lab coats are very useful because they are flame retardants, as well as keeping your skin and clothes safe from any chemicals if there are spills. If you're about to start an experiment, you always need your lab manual, not the lab notebook. And every time you're about to start something, you need to get a pair of gloves before you touch any of the glassware or any of the chemicals. Always use gloves when touching any of the glassware or chemicals in the lab. Now that I've covered labware, let's go over fire safety. If there's a fire, in the front of the lab, there is a fire extinguisher which Tara is showing you, as well as a spill kit which is shown below. If there are any spills, there is a safety shower and eye wash. The eye wash is in the back of the lab, and when you pull it down, it automatically turns on. The safety shower is located with the eye wash, and you pull down there to bring water down. There is a curtain for your privacy as well. If there's broken glass, please call your TA. They'll take care of the spill. Here you can see some glassware that has been broken. Tara is cleanly wiping it into the dustbin and making sure to get all glass. Notice when she goes to clean up the glass on the floor, she does not kneel on the floor directly because there may be shards of glass that can get into your clothing. Please do not kneel on the floor and make sure you sweep up all the glass in a wide area. Once there, bring it to the glass waste and dump it all there. The dustbin will go back in the bin up front. Safety's out of the way. Our first lab is thin layer chromatography of substances, and it's a two-part lab. The first part of the lab will be TLC of Analgesics. In this lab, we will be using TLC to determine the composition of unknown mixture of analgesic. We will be given 5 known reference substances and an unknown consisting of a mixture of 3 of the references, as well as an unknown containing a single reference compound. Here on the right, you can see the 5 analgesics we'll be working with. A quick refresher on how TLC works. So, TLC is a very simple and cheap method that can actually separate quantities of less than 10 micrograms of material. Chemists commonly use TLC to actually understand a reaction better. They can monitor the disappearance of a reactant or the appearance of a product by the spots. TLC, like all other chromatographic methods, have 2 parts. One part is a stationary phase, which is an absorbent, and then the other is the liquid phase, which actually carries the compound of interest up or throughout the stationary phase. The compounds in the sample mixture will move through the stationary phase at different rates due to the attractions for the mobile and stationary phases. The individual compounds in the mixture separate as they go through the stationary phase. So during the TLC process, the solid stationary phase, called the adsorbent, adsorbs the mixture compounds. As the mobile phase, called the eluent, travels up over the adsorbent, the compounds within the mixture moves at different rates. A reversible and continuous competitive attraction between the eluent and absorbance for the mixture compounds causes this rate difference. Compounds with less attraction for the adsorbent move rapidly with the eluent. Compounds with more attraction for the absorbent move slowly with the eluent. Because TLC adsorbents are typically very polar, the more polar the compound in the mixture, the more strongly it adheres to the adsorbent and the more slowly it moves. Similarly, intermolecular attractions between the eluent and the compounds determine the solubility of the compounds in the mobile phase. In general, the more polar the eluent, the more rapidly a given compound moves. Polar compounds, which are strongly attracted to the absorbent, require polar eluents to attract them away from the adsorbent. So we started with general safety. Now we will do safety for the TLC lab. Here is the general setup. Everything will be inside of the fume hood because the chemicals are dissolved in organic solvents, which are very volatile. Here you can see each of the analgesics are in a secondary container to prevent any chemicals from spilling. You can see everything is labeled, so please do not mix and match them. There is also capillary tubes that are found here and a waste container. There are a few other things I'd like to talk about in safety. The capillary tubes, even though they're small, are made out of glass and can stab you. Also, the TLC plates are made out of glass and covered in silica. As you can see, Tara wipes the plate, and as you can see, it comes off very readily. Silica is very dangerous. Here, I have essential safety, and I've mentioned all the chemicals: hexanes, methylene chloride, ethanol, ethyl acetate, and acetic acid. All organic solvents that are extremely flammable and will cause irritation to the skin, eye, or respiratory tract. Also, all of the analgesics are also irritants if touched on the skin, so please do not do that. Silica plates you need to be careful with because the dust, silica powder, is very dangerous if inhaled. It actually will end up causing permanent damage in the respiratory tract, specifically your lungs, because the body has no way to get rid of them. Please be careful with the UV lamp. It causes DNA damage to humans, so keep it away from your skin, face, and eyes. Now that we've gone over general safety, I'm going to talk about the experiment, and we're going to first start with preparing the TLC plates. Here you see Tara has a pencil, which is dull, a ruler, as well as a plate. And notice that she's marking a little dash on each side of the plate. This is to help draw the origin line. This origin line is important as this is where we will be spotting our chemicals. You will see later why this is so important. Notice that when drawing the lines, she is being very gentle not to dig into the plate because the silica is readily removed with hard force. That's why a dull pencil is ideal. Notice she's drawn 4 little lines. These lines are where you would spot your chemicals. Ensure that there is enough space because you do not want your chemicals to overlap, as you will mix and contaminate them. And notice that Tara is labeling them using the 3-letter and 2-letter codes found in the book. Here she is preparing the second plate. Once again, notice she is using the ruler to draw the 2 lines around 1 centimeter from the bottom of the plate. And now she is drawing the origin line. This is once again where you spot the chemicals. One thing I want to point out is that Tara is drawing enough lines to make sure that we have space for each reference standard, the mix, as well as the 2 unknowns that we will be spotting. This is important because we will need the 5 reference unknowns to help identify what is in the mix as well as the 2 unknowns. So we've learned how to properly mark and label our TLC plates, but here are a few things that we should not be doing. Tara here is using a pen to mark her TLC plate. This is very bad practice. This is bad because the pen ink contains multiple organic compounds, as opposed to a pencil, which is made out of inorganic compounds. The organic compounds found in ink will actually dissolve in the solvent that you're using. So, any of the organic compounds found in ink will then run up your TLC plate and thus in turn contaminating it, which would be very bad for your experiment. Here, Tara is marking the TLC plate with an Expo marker or a Sharpie, and that would be very bad because both of these pens essentially have ink that is made out of organic compounds. So now that we've learned how to spot our TLC plates, let's go ahead with the experiment. Tara is working in the hood because, as the procedure said, the chemicals are dissolved in a mixture of dichloromethane, an organic solvent. Notice she has her two plates in the hood and is working with the sash at a lower height. She's removing one of the capillary tubes and is going to spot it. So essentially, these are tubes with a very small inner diameter. And due to capillary force, it only needs to touch the liquid, and due to surface tension, it will climb up the tube and fill it up. Once there's liquid inside, you can do one small gentle tap on the TLC plate. Here, we've changed the angle so you can get a better view. Tara now has liquid in there, and as you can see, one spot she adds on there. And you can actually see that it has been spotted. Sometimes you can do multiple spots. However, it's ideal to do a tap and not hold it on the TLC plate. The longer you hold it on the TLC plate, the more it will spread out and form a large round spot. You do not want that, as that will give you a bad TLC reading. Here, Tara is going to continue spotting the rest of the standards, the mixture, as well as the two unknowns. Today are unknowns 12 and 25. So now we've shown you the proper way to do things. Let's show you some things that you should not be doing when performing TLC experiments. So Tara is going to show an example of overspotting. Here she is holding it down, and as you can see, the spot is getting extremely large. This is going to cause a result that shows a huge spot and smearing. Now she's going to show what happens when you spot two things way too close to each other. You can probably figure it out. If you make the spots too close to each other while running the TLC plate, they will merge together and give you a false reading because the chemicals are so close to each other they'll mix. Now that we've spotted this, we're going to run the plate just to show you what this looks like. So we're going to have that run and set it aside. We'll talk more about how to run the plates when we're going back to the main experiment. Okay, back to the main experiment. So sometimes you want to check to see if your spots are spotted correctly. You can use a UV lamp to check your spots and make sure they're not too large. As you can see here, they are evenly spaced and not too large, which is ideal for running a TLC plate. Now, Tara is going to get our TLC developing chamber. As you can see, it's labeled, and our solvent, which is also labeled. We're working in the hood because the solvent has acetic acid as well as ethyl acetate. So when pouring the solvent, ensure that you only pour a small amount to cover the bottom of the developing chamber. You need enough that will be below the origin line that you drew before. Here Tara is checking to see to make sure that it covers the bottom of the developing chamber and allows the TLC plate to stand straight. If you don't have a perfectly level line, you're not going to have even development. You'll see the solvent coming up as a curve instead of a straight line. Notice that she quickly closed it to make sure that the solvent doesn't evaporate. Now she's holding the TLC plate next to the developing chamber to make sure that there's enough solvent. So now she's going to insert the plate very slowly and to ensure that it's balanced in the center and that the solvent is below the origin line. In these developing chambers, you can run up to 2 plates at the same time. It is essential that they do not touch. They must be spaced enough that there is space in between them. Here, Tara is trying her best to ensure that that's the case. Once you put the plates in, quickly close the chamber or the solvent will evaporate and change your TLC reading. This is important. If you don't have confidence at putting 2 plates in at the same time, you can run 1 plate at a time. There is no real downside to this. The only thing is that it will take longer as you have to run 2 separate plates. However, If this prevents you from making a mistake, this is the ideal route to go. Here you can see the solvent climbing up. It's going rather quickly and quickly passed over the origin line. So a few minutes later, we can check on this plate, and then we see that the line has risen up to the top. We want to make sure that it doesn't reach all the way to the top. If it goes over, the solvent will still be carrying any molecules and then affecting your TLC reading. Notice Tara takes out the TLC plate and immediately draws a line at the line where the solvent went to. This is called the solvent front, and this is important for characterizing any chemicals on the TLC plate. Once the solvent front is marked, you can put it to dry. Here, Tara's just holding it out and wiping any excess solvent from it. Once the back is dried, she places it down to dry. Now to mark any spots on the TLC. So, now that it's done, we're going to use the UV lamp. Many of the chemicals today have our aromatic rings, and these absorb in the UV region. So using the UV lamp, we can detect many of these compounds. Here, Tara is using the lamp to draw a circle around the compounds. Here you can see the spot very clearly, and she's drawing a line with a circle around the marks. Here we've run the second plate, and Tara's going to check it again and draw the circles around the spots. Sometimes you may notice a spot that has a streak. That has to do with the way that the chemical is running up the TLC plate due to its polarity and interaction with the silica. The way the spots form tell a lot of information about it. And you can infer this based on the solvent that you're using, let's take a look at the plates that we spotted earlier that were done poorly. So, here's an image of the plate. As you can see on the left, there was one that was streaked with multiple spots, and the one on the right there are too close together. If you put too much chemicals, you'll have a smear. And in the way before mentioned, if you put them too close together, they'll mix together. Here is another poorly done plate. This has too much chemicals spotted. When you add too much chemicals, the TLC plate will smear. This is not characteristic of the compound. This is because you've put too much chemical on there. A quick review on calculating the retention factor. So before, I mentioned that the origin is very important. So that is labeled letter A. There I have letter B, which is labeled the solvent front. C and D are basically lines drawn from the origin to the center of the spot. When calculating the retention factor, it's ideal to draw the line to the center. E is then all the way from the origin to the solvent front line. And to calculate the retention factor, you're actually going to calculate a ratio of either C or D over E. So basically the distance that your compound has traveled versus the distance of the solvent front. This will give you the retention factor, a number between 0 and 1. And this will relate to the polarity of the substance based on your solvent system. Now that we've reviewed RF, let's take a look at the results from part 1. Here is a TLC plate of the 4 standards: AC, ASP, caffeine, and ibuprofen. Please take a screenshot and calculate them, or get the data from Blackboard. Here are the other 4 samples: the salicylic acid, the mixture, as well as the 2 unknowns. Calculate the RF and then compare these spots to the known standards to figure out what is in the mix and the two unknowns. Okay, now to start extraction in TLC of chlorophyll A and B from spinach. So TLC is really powerful. You can observe many chemicals in plants by using TLC to separate them. Once you grind them up physically and then spot them on the plate, based on their polarity and how well they absorb to the silica and the solvent system, you can actually separate and characterize multiple chemicals inside of them. And that's what we'll be doing today with spinach. Okay, now let's go over the safety for part 2 of the lab. A lot of the things will be the same. We'll be using many of the same solvents, specifically hexanes and ethyl acetate, both extremely flammable, volatile, and will cause irritation to the skin and respiratory tract. We'll be using spinach, which should not be eaten, magnesium sulfate, which is used to absorb water, and sand. Both can cause irritation. Magnesium sulfate can induce vomiting if ingested. We'll also be using the UV lamp, so please do not use it on your skin. Now, materials for the lab. So here we have spinach and sand, and to the left the scale, which you'll see in a moment. Now let's go into the experiment. Let's start with weighing out the spinach. Many of you guys have used this scale before, but we're going to review. So here you can see Tara is cleaning off the scale to make sure nothing's on it and waiting for the scale to go down to 0. Once the scale is at 0, she tears it to ensure it's at 0 and then adds a balance book to it. There, she closes the door and then waits for it to equilibrate. You can tell because there will be a small circle on the screen. Once there, she will 0 it and then weigh out the spinach, which should be 0.5 gram. .500 grams. Get as close as you can to the value and just ensure that you write the number down in your notebook. Tara will weigh out the other solids. She will weigh out the magnesium sulfate and the sand. Here, the procedure remains the same. She will once again 0 the scale once putting the balance and then weigh out her solids. Here, she is using a scoop to slowly add everything to the scale. Because of the size of the bottle, she's opening both doors and then using the spatula to spoon it in to make life easier. Just remember, once you want to weigh your sample, you must close both doors. And here you can see her doing that, as she weighs out close to 1 gram of sand. Once again, it does not need to be exact. Here, she's going to clean the spatula before she uses it to weigh out the magnesium sulfate. So now she will remove the sand and then get a new balance plate. Here, she cleans it and then closes to 0 it again. Now she's going to weigh out as close as she can to 0.5 grams of anhydrous magnesium sulfate. And once again, magnesium sulfate is used to absorb any water in solution, because water will affect how the compounds all run on the TLC plate. That is why we're trying to remove as much of it as we can, as well as it acts as a source to grind up the spinach. Here, now that she's finished weighing out, she's cleaned her spatula and is removing her substance. And remember, once you're finished, to clean the scale as well as 0 it for the next person. Now that we have weighed out everything, we will start the extraction procedure. Here, Tara is going to rip up the spinach inside of the hood, and then Once she rips up the spinach, she will place it inside of the test tube. When adding the spinach, ensure to pack it down using a spatula, which here you can see Tara's doing. Magnesium sulfate to the test tube, being careful to get it all in. Notice how she is using the block to keep the test tube stable, and using the spatula to place in all the sand and magnesium sulfate. Now with all the solids added, she will use the glass stirring rod to help grind the spinach. Remember to use the wooden block to ensure that you do not break the test tube, and do not apply too much of a force. If the glass rod does not work, use a spatula as it is finer and can mix better inside of the test tube. Here you can see Tara is gently mixing everything, and as you'll see, it'll break down the spinach. After grinding it, it should be a homogeneous mixture that's green, and then using a graduated cylinder, measure out 2 milliliters approximately to add to the test tube. In order to add it, you can either pour it directly in or use a pipette to add 2 milliliters into it. Once the 2 milliliters of acetone are added, you can mix it either using the glass stirring rod or the spatula that you have. Make sure to mix it in the wooden stir block or gently stir it in your hand in order to ensure proper mixing. You will notice a slight green color being taken up by the acetone after just a few seconds of mixing. Continue to mix for at least 2 minutes before sealing the test tube and allowing it to sit for 5 minutes. After waiting for 5 minutes, check on your mixture. You should notice that the acetone solution has become a much darker green color and that all the solids have settled to the bottom. Using a pass-through pipette, remove the top layer. So only remove the liquid and put it into a clean test tube, which you can see is on the right. Try your best not to take any of the solid particulates with you. If possible, stuff the pass-through pipette with cotton. Now you should have a solution that's a brilliant emerald color. Prepare your TLC plate by drawing the origin and marking a spot. Using a capillary tube, try your best to get the spinach solution in the tube. You may also use a watch glass and pour some of the solution out. Once there, spot your TLC plate. Try your best to add a few spots without oversaturating the area. Here, Tara is marking 2 spots. Remember, you can always use the UV lamp to check if you have spotted enough. Once you've spotted your plate and checked that there is enough sample on it, ensure that your jar has enough solvent in it. Remember, we are using 70% hexane and 30% acetone solution for this experiment. Check the TLC plate to make sure that you will not go over the origin line once placing it into the developing jar. If you pour extra solvent in the developing jar, get a beaker and pour it out, and then dispose of it later. Remember, once placing the plate in, be careful. Ensure that the solvent does not go above the origin line. Once settled, quickly cap and wait a few moments. Make sure to watch your TLC plate so it does not overdevelop. After a few minutes, Tara is taking out the TLC plate. Note that she's only touching the top and going to quickly draw the solvent front line using the pencil. Now that we've developed the TLC plate, let's take a look at it. Here you can clearly see that these spots have colors. That's very important when characterizing these compounds. Here I've helped identify these spots by circling them and increasing the contrast. Take note of the color and the spots to help characterize them. Remember to take a screenshot or get the data from Blackboard for your analysis. Now for lab cleanup. Any of your glass waste must go in the glass disposal. That includes TLC plates, capillaries, and Pasteur pipettes. Gently place it into the waste bin. Do not throw them. Weighing boats can be directly thrown in the trash. When cleaning any glassware or any spatulas, please use a scrubber brush and Alkanox, that is soap, and use DI water to ensure that there is no residue left behind. Only a small amount of soap is needed. Ensure that you scrub the glassware and any of the spatulas thoroughly and rinse them at least 3 times before putting them to dry. Once they are dried, you can place them back where found.